Our next speaker is Surabhi Kokane from Stockholm University in Sweden. Surabhi will share the cryo-EM structure of the NHE9 homodimer and its interaction with the endosomal lipid PI35P2, right? This lipid enhances sodium binding, offering new insights into NHE9's role in regulating intracellular pH and its relevance to disease. So, Surabhi Kokane is a PhD candidate in David Drew's lab in Stockholm, Stockholm University, where she focused on the structural and biochemical characterization of membrane protein transporters, particularly solute carriers, SLC 989. She holds a master's and a bachelor's degree in biotechnology from the Institute of Bioinformatics and Biotechnology in India. So, welcome.
Thank you, Nanan, for organizing this user meeting and inviting us for the meeting. So I will be talking today about this PI35P2 mediated oligomerization of endosomal sodium proton exchanger NHE9. So coming to the exchangers, so sodium proton exchangers are one of the Asian exchangers which are also seen in bacteria. And in the bacterial exchangers usually transport two protons in exchange of one sodium, and they are always electrogenic. Whereas in case of mammals, these exchangers are usually found to be electroneutral, where they transport one sodium in exchange of one proton. So in mammals, there are 13 sodium proton exchangers found. They are classified into SLC9A, SLC9B, and SLC9C, where the SLC9A is subdivided into two families. The ones which are from NHE1 to NHE5, they are plasma membrane localized. Then NHE6 to NHE9, they are endomembrane localized. And then the SLC9B family is very similar to the prokaryotic exchangers. And SLC9C is sperm-specific sodium proton exchanger, which has an extra voltage sensing domains. So usually this sodium proton exchangers, they have they are like a homodimer, where they have a scaffold domain, which is like kind of static, and then there is a dimer domain, which kind of glides towards anchoring to the scaffold domain. So here's a beautiful movie from a bacterial sodium proton exchanger, where we had the structure of, oh, I think it's not, yeah, I think it's not working, but it transitions from an outward open state to an inward open state. Okay. So usually NHEs from mammals, they have a long cytoplasmic tail where lipids and some protein bind and regulate these transporters. And the previous known NHE9 structure, it had 13 transmembrane helices, of which six were dimer domain, the other six were transporter domain, and there was another helix which was connecting these two domains called as TM7. Then another NHE one structure was predicted, and it was bound to a complex. It was obtained in the complex with CHIP1, and this CHIP1 was regulating the activity of the transporter. So when the transporter was expressed in the plasma membrane, it had a very low activity, but when CHIP1 was co-expressed with NHE1, then the activity was enhanced. So this protein helped in a regulation of the sodium proton exchanger. So specifically in NHE9, there was this loop region between the TM2 and TM3, which was unresolved, and it had some positively charged lysines and arginines in the region. And we were curious about like what exactly this region looks like. And we were trying to understand like how this can be resolved using cryo-EM, and we can see the architecture and if this loop domain is regulating any activity of the exchanger. So in the previous paper that we had, we did some mass spec for NHE9. We purified this protein with brain lipids because NHE9 is usually present in brain cells. So we purified this protein with the brain lipids, and we found 1 kilodalton adducts bound to this protein, and which like uh, this mass would correspond to a molecular weight of a pip2. So we thought, okay, maybe this lipid which is bound is pip2. So we did this thermostability assay where we take the purified protein and then we heat it at different temperatures and try to measure the fluorescence of the protein. And as the temperature increases, uh, you can see how your protein is stabilized when it's in the presence of a lipid. So in this thermostability assay, we tested different lipids, and we found that it was mostly stabilized by pip2 and pip3, and somewhere with DO like the PA. So we were trying to get a structure of NHE9 with pip2, but we were kind of unsuccessful with that. So we un- we wanted to see if this pip2 where this pip2 really binds. So we tried to mutate some of those lysines which were there in the loop region, and then we again tested the thermostability assay, and we found that this loop mutant was not stabilized in the presence of pip2. So we were sure that okay, this site could be a pip2 binding site. So so the because these sodium protein exchanges are too dynamic, we try to purify this protein in a lower pH. So this would make the exchanger less dynamic, and we could actually get a state where we could resolve the structure of the loop domain. So we tried to do this, and we could actually collect few particles where we could see in the 2D classes, like there's some flexible loop. We extracted those, and using 3D variability analysis, we did some cluster analysis, and then we refined those clusters, and then we picked out 42% of the particles which had this loop density and tried to resolve the loop domain structure. So in this structure, we could actually see the loop domain, but the TM regions were poorly resolved. So you need if you have a focus mask around the TM region, then your loop region is not well resolved. But if you have the mask around the loop, then your TMs are not so well resolved. So we wanted to see the loop domain structure. So we focused on the loop domain, and we could resolve the structure. And with the AlphaFold, it already predicted that the loop domain has a with okay confidence score. So we tried to fit this loop domain into AlphaFold model into the loop domain of the obtained structure. So we could see that this density clearly fits into the maps that we got for the loop domain. And we could see that when you put it into a dimer, it kind of flips, and one of the loop domain of the protomer flips to the other protomer, and it forms a nice beta-hairpin loop above the dimer interface. So we tried to solve the structure, and we could see that the positively charged lysines, they were actually pointing to at the dimer interface. So we thought that, okay, this could be like a pip2 site, and we wanted to resolve the structure of this. So another strategy that we used was adding cysteines and locking the protein into inward open and outer open state. So we tried to do that, and also we enhance our purification with the brain lipids. So we could see that now the 2D particles have the density for the loop domain and also some density for the C terminal for NHE9. And after resolving at the structure, we could see that the cysteines were not actually trapped. So maybe because of the lipids, the structure was better resolved, and also we could get the lipids at the dimer interface. So we could clearly see T-shaped densities at the dimer interface, and if we try to fit the lipid there, we could see that it's only Pi3-5P2 which is fitting in the density, and if we add the PIP2, the Pi4-5P2, it couldn't fit into the density. So then we were sure that, okay, this the lipid that is binding at the dimer interface, it's Pi3-5P2, and when Pi3-5P2 is bound, these the loop domain lysines, they cluster towards the dimer interface, and now the surface is more negatively charged. So this would allow easy access of ions to the protein. Further, we went down to checking the localization of NHE9. So NHE9 is primarily localized in late endosomes, and also the if you see the expression of PIPs in the endosomal pathway, you could see that it's prominently Pi3-5P2 is found in late endosomes. So that also confirmed that, okay, the lipid that is bound to the structure, it's Pi3-5P2. So further to test and confirm with the lipid specificity, we did this GFPTS assay where we take the purified fusion, then we add those lipids and incubate the protein for 30 minutes, and then you add the detergent beta OG, and then you heat the sample for 10 minutes. And after heating, you spin the sample for around like 45 minutes, and you measure the fluorescence intensity. So we could see clearly that the the apparent TM that was obtained for NHE9, that was around like 51 degrees. And in presence of lipids, the TM shifted to with Pi4-5P2, it shifted to 59 degrees, and with Pi3-5P2, it shifted to 66 degrees. So if you compare the purification of NHE9 when you have the dimer, and when you see that this loop is mutated, it completely shifts to a monomer. So we could see that, yeah, this lipid is also stabilizing the dimer. And if you heat the sample at 50 degrees and again inject on FSEC, you could see that Pi35 has a 35P2 has a better stabilizing effect than Pi45P2. And we took this lysine mutant and tried to dope it with the lipids, and we could see that there's no shift to the dimer. So it was mostly monomer. Then to again check the activity of NHE9, in presence of these lipids, we did solid state membrane electrophysiology. So in this, we use this rapid dilution method where we take the East polar lipid liposomes and we incorporate the protein into the liposomes with adding sodium cholate, and then do PD filtration. And then you have this proteoliposomes. And these proteoliposomes are added to a sensor, and then we use choline chloride as a non-activating buffer and sodium chloride as an activating buffer, and try to measure the signals for NHE9 activity. And we could clearly see that with increase in concentration of sodium, NHE9 showed that it's active. And then when we have Pi3-5P2 with the protein, it was more active. And the binding affinities that we got for NHE9 star, like which is without the lipid, was 36 millimolar. And when adding the lipid, it went to 9.6 millimolar. So it clearly showed that when you have this lipid, protein is more active. Further, we also tested the loop mutant, and we could see that even after adding the lipid, it still couldn't get higher activity. So from this, we confirm that the lipid is bound to the dimer, and the dimer is more active, like because these are negatively charged patch around. the outward facing surface, and that attracts the ions to the protein. Further in the structure, we could also see that because this was well resolved, the ion binding site has this sword bridge, which is arginine 408 and E239. They form a sword bridge, and that kind of uh, keeps the ion binding site more tighter and binds that can help binding the sodium in a more tighter fashion. Uh, Also, because we could resolve the C-terminal region of NHE9, we could see that the TM7 has a lysine which is directly interacting with the C-terminal domain. And this is, we hypothesize that this could be a part of autoinhibition mechanism where you have this C-terminal domain which is blocking the transport. But when you have any protein with any complex, the C-terminal domain is moved away, and then the transporter is more active. That's why we could see that NHE1, the transporter had more activity compared to when it didn't co-express with CHIP1. Further, to validate this, we also did some MD simulations. So these simulations were run for around 500 nanoseconds, and we also tested the mutant and also the lysine, like which lysine is directly interacting with the loop with the lipid, and we could see that the lysine 105 was making contacts directly with the lipid. So, and also in the presence of PI35P2 and 45P2, we could clearly see that PI35P2 had a better stabilizing effect, and it was better fitting into the density compared to PI45P2. We also did some cell-based assays in collaboration, and they could clearly see that when they express NHE1 and NHE9 both to a plasma membrane, NHE1 is active and NHE9 is completely inactive, and which makes sense because PI35P2 is not present in the plasma membrane, so the protein is not active there. But when you express this protein to endosomes, it becomes again active because now you have the lipid there around. And we also tested the pH change, and we could see that the pH drops from 5.5 to 7 when you have this protein expressed to the endosomes. With this, we propose a functional model for NHE9 where when NHE9 is expressed in plasma membrane, there is no PI35P2, so the transporter is less active, and it's because it's more dynamic and the loop domain is not stabilizing the protein. But when you have it in endosome, because there is a lipid present, this lipid stabilizes, and then the transporter is more active when you have it in endosomal membrane. With this, I would like to conclude my talk, and I would like to acknowledge all the lab members who have supported me and during my whole PhD, and also collaborators, Michael, Carol, Daniel, and Lucy, for their uh, experiments and whatever support that they have given to us. And also, I would like to thank my supervisor for giving me a chance to talk.
Thank you so much. Thank you, Surabhi. Questions?
Thank you for your talk. I would have a couple of questions on the electrophysiological measurements. So the first is, I saw that you jump with sodium chloride and with pretty much high concentrations. So did you also implement some empty partial liposomes as control? Because when you jump with charged substrate on SSM, you can generate non-specific currents.
Yeah.
So did you also check for this?
Yeah, so for this we had a dead mutant. Like if you see, like the currents for the dead mutant, it's the background is too low. And also we had another protein which we use as a background to test if it's not giving a high signal. Yeah.
So at this point, did you also, so basically you use this as a proof that your protein is right side out in orientation in your proteoliposomes. So you didn't check with other
No, we didn't check orientation because it transports in both directions. So for us, the orientation is not a concern.
So it's not, yeah. Thank you.
Thank you. Another question?
Thanks for the very impressive story and the nice structure. I have a question regarding this SSME assay. So do you know what's the potent sodium stoichiometry metric of this NHE9 transporter? Because in E. coli, this sodium potent antibody NHAA, and that's two potent exchange for one sodium. So indeed, in the SSME assay, we see the efflux of positive charge that should be give a negative response. But here I saw a positive response.
Yeah. So this is 1:1 stoichiometry. That's why you have the positive signal. Yeah, 1:1. So these are electroneutral transporters. So it is having it's not two protons in exchange of one sodium. It's always one sodium and one proton.
So just I don't understand. Okay, if it's electroneutral, so why we say the positive response here?
I think for electroneutral you see a positive one. If it's electrogenic, you always see a negative thing. So
Andre, you have a comment?
Yes, so I think in that case you see a positive response because sodium translocation is just faster than the proton translocation backwards after. So you can distinguish by the kinetic properties. Sodium translocation. If one is faster, you can see it. I actually also have one question. You are showing KD values there.
Yeah.
How do you conclude that you obtain the KD?
We did this LPR ratios, and with the increase in protein concentration, like you see faster decay. So we tested the LPRs and we could see that the decay was same for the LPRs.
So yeah, that proves that you actually see a pre-steady state current, so no steady state,
Yeah.
which anyway couldn't be measured because it's an electroneutral overall transport. So just the comment that it's actually not a strict KD because KD is the binding of the sodium, but it's actually the EC50 of the process you record, and that's the half cycle of the transport. So it would be better to mention that it's an EC50 and explain what that means. It's probably closer to a KD than to a KM, but it's still not a real KD.
Yeah, I mean, I agree with this, but and also like then there are some papers on NHE1, NHE2, NHE where they measure the transport of sodium with the radioactive sodium, and they have seen that the Km for sodium was around for NHE1, NHE2, and NHE3 it was around 10 to 15 millimolar. So this KD values were like very close. So we kind of put this as a KD, like with a previous number.
Okay, thank you very much. Big hand for Surabhi again. Thanks for coming here. And also for people tuning in just for the surfer session. Ulrich Hammer's talk has been moved to previously today. So if you wanted to see this, just reach out to us. Thank you very much again.